Okay, sorry about that. My computer was like, you don't have enough disk space. It's because I don't. I have a external drive connected, so we should be good. Oh, turn this off. Okay, I don't need to hear myself talking. Okay, let me turn a few things off here so everyone can see what I'm doing. I promise you I've been working extremely hard. Get all this together. So this thing does not like to connect to the chat. Yeah. Okay, there is some people here. Fantastic. My chat is not connecting right now, so I apologize if that's true for you too. I may exit out and try another. Don't worry, I'll keep streaming, but I may exit out of this tab. So you'll still be able to see me. I can't see your comments right now. I am still streaming though. So let's go quickly to Twitch. Okay, see if I can get it back on. And I'm here, just trying to make Twitch load its chat for me. Okay, good news. It's loading the chat, and I'm still live. Fantastic. Oh, okay. I blitzed out for a second there. Okay, we're all good. All right. Excellent. Hello to the couple people I see in the chat already. That's not everybody, but it's good. Again, this weird person watching me. Pretty sure they're just some sort of lurker. Okay. We'll get started in a few minutes. I have already posted this. Trying to get everything a little bit more ahead than I have in the past. Probably going to be behind by next Monday, but we're going to see what happens. All right. So it's a show. Okay. I think I'm wrong about the chapter. I think it's chapter 14. We're only doing the very first part of it, too. So let me type that in, actually. 14, 1, it's 14.1 through 14.3. That's all we're going through. We're not going through that much of 14, actually, because it is a large chapter that covers a lot. We may come back to it a couple times before, but mostly I wanted to cover the basics here, okay? Again, there's a lot in this. All right. This is not material on the test. No, chapter 14 is all new. And again, chapter 13 is only like, how would the ribosome read stuff? And if the DNA says this, what would the ribosome read? So that's the basic side there, okay? Don't stress too much about that. Everything is mostly 7 to 12, with only a little bit of 8 and a little bit of 13. So focus on those parts and you should be okay. All right. Okay, well, this started just another minute or so. I've been working hard on the overall test. Sorry, not the test, on your case study for next week. Your case study is interesting, and I was trying to structure it in a way where you are not going to be mired in like 5 scientific papers like it was set up. So instead, we're going to be focusing mainly on the diversity of gene mutations for cystic fibrosis, which will be interesting, I think, especially after we go through this chapter. All right. We'll get started in a minute. 5 viewers right now. It's okay if you're a few minutes late. Oh, 6. Great. Okay. All right, I'm going to stop watching the number of people come in. So there are people coming in. Our class is starting soon. Okay, gene mutations. Again, I have not posted your case study, but it is just about done. I wanted to edit one more thing, and then I'm going to send it out, so you'll be able to see that. Again, worry about the test right now. It's not really that important if you have the case study done. That's not due till next Wednesday, so hopefully you'll still have enough time. Again, it will be posted very soon. Your homework is posted. It's on gene mutations, what we're covering today, and probably a little bit of Tuesday. And then we're going to start going over so we've talked a lot about functions of gene expression. And in chapter 15, we're going to start going through actual bacterial and eukaryotic gene regulation. And then finally, we're going to focus in the last part of class on actual genetic engineering, which we have been doing a lot in the case studies already, but I wanted to clarify and have a segment in the lecture as well. All right. So let's get started. One more check for, all right, good. We have a good cadre of people here. Okay. All right, gene mutations. So we talked a lot about kind of how genes are read and expressed. We're going to talk about mutations now. So mutations overall, they're heritable changes in the DNA. Okay. Ebere has and Aguero, Faye has already covered a lot of the stuff that we're going over today, but we're going to go in more detail about now that we've covered a lot of the pieces, how exactly gene mutation can express genetically and express in your body. Okay. So you can have a gene mutation, a somatic mutation, okay, mutant somatic cell, and then as mitosis happens, that can spread. An example of a type of mutation that happens in your own body is moles. Got one myself here, okay. So this mole is a mutation of a skin cell. It just shows up as a little tag. It clones itself. It has deep melanin. It's a type of mutation in your somatic cells. Okay, you have some probably. Those are, and too much mutations, and sure, you could get cancer stuff. This is why I should always pay attention to what my molds look like. If they're nice and round and small like this one, then it's not a big deal. But those mutations in your cells can also become cancerous. Germline mutations are when the actual offspring carries a mutation, okay? So germline cells are ones that turn into eggs or sperm, and through sexual reproduction, about half of the generation will get that mutation because of the way that meiosis happens. And sexual reproduction and people who don't have the mutation, okay? So we're going to be talking about the types of gene mutations based on their molecular nature and then on how they express in the body, okay? So base substitutions, nucleotide point substitutions, insertions and deletions, and something else that's interesting, the expanding nucleotide repeat, okay? I'm not going to explain how that one happens. That's actually a pretty interesting one, okay? So base substitutions, there's transition and there's transversion. I'm going to talk about both of those. Frameshift mutations, that the insertions and deletions can be, we talked about this a little bit with muscular dystrophy, is that a frameshift mutation can change the entirety of a protein by shifting over this. I've talked a little bit about frameshifts before, but we're going to go over that in detail today. And then expanding the nucleotide repeats. So this one is an interesting process by which it happens. So I'm going to walk through why this would repeat over and over. Okay. All right. one thing to know is that mutations can occur anywhere, and sometimes they don't make a difference, and sometimes they do. Okay? Mutations can occur in coding or non-coding regions. They can occur in regulatory sequences. They can occur in promoters. They can occur in enhancers. They can occur in the splicing signals that says where do you splice the introns and exons. All of those can cause the RNA transcript at the end to differ from what it's supposed to be. And depending on how it differs, it may or may not cause a detrimental mutation to occur. So it's sort of a weird piece. Now, this is the overall process of RNA polymerase reading the entirety of a gene. Okay, so again, you have the ta-ta box right here. RNA polymerase attaches to, using sigma, that attaches to the promoter, reads and transcribes the promoter, and reads the coding region, and then has the terminator. Okay, so this is the whole thing. The promoter is on the RNA transcript. The RNA coding region is on the RNA transcript as well. This promoter is cut out of mRNA. Okay, but anywhere that has to do with the recognizing and coding of a certain region, all of that is included in the overall what it's going to be translated into. Okay. All right. So besides the ta-ta box, okay, which is and the promoter, which are upstream of the coding region, even further upstream are these small enhancing sites. If there are activators, then they will bond onto this protein complex, okay, and to the enhancers, sort of wrap it around. If there's a silencer, a repressor, it would do the same thing, and it would make the activators go away, which means the RNA polymerase would not attach or would not transcribe after it attaches. It would just tell it to go away. So you have these interesting pieces. Any one of these can impact the presence of, the frequency of, and the quality of the RNA transcript. If a mutant occurs anywhere that influences the transcription of a gene, erroneous or completely missing proteins can occur. Note that a missing protein is just as bad as a erroneous protein. That missing protein means that you have a that completely lost protein, okay? And the lost protein itself means that that this would act like a non-functional gene here. Any of the ones that encourage the RNA polymerase to attach would cause a missing protein, okay? So we're going to talk about how mutations impact proteins in particular. They alter the DNA and the subsequent effects on the protein. All right. So I mentioned this already, base substitution, nucleotide pair substitution, okay? Insertion and deletion, and those causing frame shifts, and we'll talk about other ones too, okay? So I'm going to show you this mutations, thing. Now, here's the deal. It's hard to do sound through this, but I think it is helpful to watch the cute animations, and I'll put the, this is only going to be like 5 minutes, okay? So, let me see if I can get to it. Okay. Let's get a copy of that all together. Here we go. All right. Just gonna pull this over. Oh, you don't want to do that. Okay, so it's in my recent searches, right? There we go. Okay, we'll get there. See if I can keep this on. You're not going to be able to hear this much, but that's fine.
With Benefiber, you'll feel the power of gut health confidence every day. Benefiber is a 100% natural prebiotic fiber. Benefiber, trust your gut.
Okay, not that. All right.
Ever since I was a kid, I have been curious about mutations. I think certain movies may have had an impact. I used to imagine mutations with amazing abilities, but there was also a lot I didn't understand about mutations. Let me explain. Many people understand that a mutation is a change of genetic material.
So we're just going to look at
more
the captions
specifically,
for now.
a change within a nucleic acid. RNA and DNA are both types of nucleic acids. Therefore, anything with RNA or DNA can have a mutation. That means animals, and that definitely includes humans, also plants, fungi, protists, bacteria, archaea, they can all have mutations. Additionally, so can viruses. Many mutations can be neutral in effect. You can see this codon codes for the amino acid leucine. But if it experiences this particular silent mutation, even though a base has been altered, it still codes for leucine. It did not change the amino acid. Mutations can also be harmful or helpful, but it's important to understand that mutations are random. The organism can't will itself to get a certain mutation. You can learn more about this in our natural selection video, where we talk about how a bacterium may already possess a helpful mutation that allows it to survive an antibiotic. But the bacterium didn't will itself to mutate to develop this certain mutation. Okay,
Right,
so
we're
again,
going to talk a little bit about bacterial mutation and
can be factors that make mutations more likely to occur. External factors like certain types of chemicals or excessive radiation, or internal factors like an event that causes a problem during DNA replication in interphase. During interphase, cells can replicate DNA before they divide. So let's discuss some different types of mutations. We'll start with gene mutations. DNA makes up genes.
We're going to only
and
talk
genes
about the
can
gene
code for
mutations,
proteins that
so
influence
I'll
different
stop it
traits.
after it gets
So
to
when a
chromosomes.
mutation
We've
in
already
DNA
talked about
happens,
chromosomal
which
mutations, so
specifically
we're kind of
means a
going the
change in
other
one or
way
more
from
DNA
macro
bases,
to micro.
then different proteins can be produced, which can affect an organism's traits. In this example, we have fruit fly DNA. Mutations could include substitution, which means the wrong base is matched, insertion, which means an extra base or bases are added in. There is also deletion, which means a base is removed. Insertions and deletions have the potential to be especially dangerous. Why? Remember in protein synthesis how we talked about how bases are read in threes? Well, if you add a base or remove a base, suddenly the number of bases total has changed. And if you read the bases in threes, depending on where it happened, everything that is read afterwards could be affected. We call this a frame shift mutation. If you look at this frame shift example here, where a single base has been inserted, you can see how the following codons, which have 3 bases each, are now all affected as the reading frame has been shifted. This example shows how it can lead to many amino acid changes. There are also chromosomal
I know you can't
mutations.
hear this
Remember
completely,
that
but this is
chromosomes are made up
I think it's
of
a fun
DNA
little
and protein, highly
animation that I can't do
organized,
on the screen
and they have
for
lots
you
of genes
necessarily.
on them. The human chromosome
All
number
right.
is 46.
Let's stop with this
23
one.
from an egg cell
Again, we've already talked about the flipping or the errors and such, so we're going to quit this now. But that gives you an idea of some of the pieces that we're going to be talking about. Again, we're going to talk about them in a bit more detail than what this has, okay? All right, but let's do this again. But here I have shared a small GIF. We have the insertion, get this new guy here, deletion, someone just disappears, or substitution. Same number here, but one of them has been flipped out. Okay, we're going to talk about those substitution pieces. All right. So we'll talk about this. Here is that the types of mutation. Okay, the and then end. Okay, as the man saw the dog hit the can, and then it end. Okay. But this is just the point mutation. One single flip, you have a change to a T. Maybe an entire word is deleted. maybe an entire word is inserted, or maybe a small part is, and then you shift it, and now every word is still 3 letters, but you've made it nonsense, okay? So we'll talk a little bit about the substitution or the point mutations, okay? So this is where one codon is changed. The ways that this can happen are twofold. Number one, you have a transition, okay? Transitions are purine to a purine. So this can mean that A can turn to G, G can turn to A, or a pyrimidine to a pyrimidine, T can turn to C, C can turn to T. That's more like a flip, okay? So if instead of A on one side and G on the other, it's G on one side and A on the other, okay? So it's possible that it just flips. Or the, sorry, I guess G and C are the bonding ones, but the idea is that that can change, or it can do a transversion, a purine turning into a pyrimidine, which is a little bit rare, but that can happen, okay? That purine instead is read as a pyrimidine, okay? A to C, A to T, G to C, G to T, etc., okay? But all of them ultimately end up with a single substitution on the template strand that the RNA is reading, okay? Meaning that the RNA is going to look different. So this pair substitution, again, it's a replacement of one nucleotide and its base, because otherwise you'd have a mismatch. Now, often what happens is that there is a mismatch. DNA comes through and it rereads, and sorry, I have this set up to play timing because I don't want to play timings. So DNA comes back through and it rereads this, and sometimes it corrects for an error. But if it corrects and it replaces the wrong one, it replaces the one that was originally there and leaves the other one, then that can cause this nucleotide pair substitution. Where this occurs in the gene really depends on its impact on the resulting polypeptide and which one gets changed. This can be reasonably benign or reasonably small in its impact on an organism. So silent mutations were mentioned in the video. Many amino acids have 2 to 3 different codons which code for the same one, okay? So if here you have an A instead of a G, this still, GGC, GGU, same thing, okay? So again, you have your template strand on the top here. And it's going to still read it the same way to code for the same one, okay? So there's a difference between a neutral mutation and a silent mutation. A silent mutation is a form of neutral mutation. But a neutral mutation itself just means it has no impact on the organism. Neutral mutations can occur in introns, or they can occur in a part of the DNA that, like in the telomere or other parts that don't matter quite as much. And that's also a neutral mutation. Silent mutations specifically change, they change from one amino acid codon to another amino acid codon of the same type, okay? So that's the difference between the silent and neutral. Neutral can be anywhere that doesn't have any impact on the polypeptide, while silent specifically does change the code that codes for proteins, but it does not change the actual amino acids. Mutations like this, though, result in no phenotypic change in the organism. You probably have some of these in your body. Remember how the genetic code is redundant. It's degenerate. It's partly because we get these random changes all the time, and as long as it's on the last one, nothing bad happens. Now, another one is called a missense. If a substitution would cause a new amino acid to be chosen, this is a missense, okay? So a single change in a single one, you have a GAG, mRNA would read GCUC, you get leucine, well, change that A to a T, flip it over, okay? So this would be the transition one, okay? GTG, mRNA, you get CAC, okay? And that CAC turns into histidine instead. So you have a completely different one. Now this is only one single change of a single amino acid. That's a little bit less. It can be destabilizing to the protein for sure, but it likely will still end up with some functionality, okay? Sickle cell anemia is that kind of missense I just mentioned. It takes the TNA and it flips them, okay? Changing a GAG glutamine into a GUG valine, okay? And that loss there changes the shape of the protein and you get sickled cells instead. A nonsense mutation is a little bit more impacting. So you guys just did the class activity, which I'm getting to the grading of. But you guys just did the class activity of the muscular dystrophy. Why was the exon skipping helpful to the helpful in particular to the muscular dystrophy? Who can remember that? All right, a bunch of you guys are here, so who remembers what? So it lowered the symptoms, yes, but what I'm asking you specifically is what was causing the muscular dystrophy gene to be so messed up? What was one of the biggest issues in the exon that they're trying to skip? So yes, the exon skips that exon. What was in that exon? Why skipping it? What did it do to the protein overall? So Beatrice, I'm going to slightly correct you here. Many proteins were being produced and they were smaller than normal. Not that they weren't being produced, is that they got cut off too soon. Why did it get cut off too soon? Why was it smaller than normal? What did it have in the code, the mRNA code in that exon? There you go. Okay. Ifanya has it. Premature stop codon. That's what a nonsense mutation is. When an amino acid codon is replaced by a stop codon, this can be much more destabilizing, okay? So this would change a normal amino acid into a stop, and this can cut off the entire production of a protein. UAA, UAG, and UGA are the stop codons, but UAU or the UUG, those are not stop codons. So if a single stop codon is in the wrong spot, the ribosome stops reading it. And that's very important to remember, okay? That no matter where the stop codon is, it will stop at that point. And since AUG doesn't occur later on, it's not like it's going to continue on later and finish the protein and then stitch them together. That's the only chance it's going to have. That mRNA is done now. So this is one of the issues with the nonsense mutation, is that it can cause how much the resulting protein is impacted depends on where the stop codon is. If it's right before the old one, you have a smaller but still reasonably functional protein. If it is far upstream, maybe you have a missing or extremely truncated protein. And that's one of the issues with muscular dystrophy. Why it is so impacting is that it's not that you're missing one or two amino acids, you're missing most of them. And that's why the exon skipping prevents that stop codon from being there, and you actually have a more functional protein because you have more of the amino acids present, and at least it can be slightly functional. All right. This can cause disappearing proteins. Again, these aren't all bad, but often when you see the lack of something, it can be caused by a nonsense mutation where the actual protein itself just isn't produced or is produced in such a small form that it no longer is functional. So this would be the purple protein disappearing here. Perhaps the purple protein is caused by a nonsense mutation where the protein itself is mostly gone or disappeared or cannot produce color because it's too short. Insertion and deletion mutations. We're going to spend a lot of time on this one too. So this is additions or loss of a nucleotide pair in a gene. So if you add one, suddenly it's going to read it TAA. It would normally read it TAA CTG. Okay, so this is in the original. Is this DNA or RNA? Who can tell me? Is this strand DNA or RNA? Okay, it's DNA. So originally, this would be read by the RNA as U. Sorry, this would be A, U, U. Okay? And then it would read it G, A, C. But you add one, and you get the first amino acid right. And actually, the muscular dystrophy is more than just a premature stop codon. It is a frame shift near the beginning of the protein. And that frame shift, what it does is it replaces its extensive missense. You have replacement after replacement after replacement after replacement, and that builds up into a completely non-functional or a destructive protein. Okay, so it's often more disruptive than NPS due to this frame shift change. Okay. So again, base insertion or deletion. Okay, so an example with words here. Okay, normal, a substitution would be it goes from beast to feast. Okay, same length long. The insertion, you could change the breast. Deletion, change the best. But the concept here, though, of the frame shift is really important. Okay, because if you insert a base or change a base, you change the reading frame in the actual RNA. Okay, so there are 3 potential reading frames for a given RNA sequence. Remember, I said, oh, I forgot that I don't actually know the answer to that question. I figured it out. The reason there's 3 different frames is that it depends on which one you start at. Okay, so if it starts here, you can either read UCA to start, you can read CAU to start, or you can read AUG to start. And that actually, the reason that this start codon is important is to get you in the right reading frame. There are a few nucleotides in an mRNA before, so there's the untranslated region, 5' untranslated region. So you have to find the actual start of the gene, and that's usually AUG. Okay, so this gives the right frame to it. But if something is edited, and actually this AUG has something deleted in it, it's not going to be able to start at the right spot. It may start way down the line or not start at all. So that reading frame is very important. I like to show this one. So this person, so obviously you can't send this text anymore because you're not going to a restaurant. But hey, I'm going to Barney's at 12. You want to come? Your mom says, sure, I'll see you there. If you remove the H in this, but you keep the same reading frame, in that the same words in the same organization with the same punctuation, suddenly this makes no sense at all. And this is would mean that you have a completely make sense to a not make sense at all. And likely if you send something in the reading frame. Now, if you send something without spaces, it's different. The key here is that the reading frame has shifted, making nonsense instead of making a coherent protein, coherent sentence, which is basically what a protein is. All right, so this is from your book, another explanation, okay? So change of one letter is just a substitution. The deletion, again, changes the whole reading frame. So does insertion, okay? the cat saw the dog changes to nonsense in either one of these because a single deletion or a single insertion. Now, if you change the G, if you inserted something there, it would be less disruptive. So it does depend on where the insertion and deletion happen. Again, this is extensive missense afterwards. Instead of having one missense, and that's kind of the sickle cell anemia, you have one broken triplet, that one broken codon causing one change in amino acid. The extensive missense can cause even more destructive events. All right, so again, this is the same thing. Talking about the point mutations, it can be either silent missense or nonsense, and those frame shift alter multiple pairs. So I already said this a couple times, so I'm not going to have you answer this. But again, a frame shift would be much more deleterious, especially if it's near the beginning, if it's upstream of a lot of the amino acids. Okay. Deletion of certain things can cause misfolding of the proteins. So cystic fibrosis, the most common mutation is that a deletion of 3 bases leads to a protein without phenylalanine. The protein therefore doesn't fold properly. So the goal, the cystic fibrosis pattern, again, this was in the previous, the study we went through on Tuesday, that this is a chloride ion, it's a chloride ion channel, okay? With the wrong folding, it does not transport chloride ions properly, even though it goes in place, and you have a buildup of mucus in the lungs. Now, this is the most common mutation, and again, we learn about this in simplicity at the beginning. If your mother has a CF mutation and your father has a CF mutation, they pass it down to their daughter, then they can not have it and their daughter can have it. We do those basic Punnett squares, but it's not always as simple as that. More than one kind of deletion can cause this disease, even more than one kind of mutation. Even some of the missenses can cause this. So this is a picture of the CF, cystic fibrosis gene, is called the CFTR, okay? It's the cystic fibrosis transconductance protein. I forget exactly. Transmembrane protein. It stands for something. The concept here is that it's called CFTR, and it transcribes, and then it comes up with this working protein. The concept here is that it's made of several different segments. Okay, you have this membrane spanning domain 1. Okay, so this is the channel here, right? This would impact the channel. So you have this membrane domain 1, membrane domain 2, nucleotide binding domain 1, regulatory domain, this other nucleotide binding domain, and all these signal transduction and cytoskeleton connections. So any of those regions can impact this. Actually, I have a friend. She does not have cystic fibrosis. She's my same age. She has survived until now. She has what's called a CF similar mutation. Hers is a mild version of cystic fibrosis. She has trouble with her lungs building up things. She's at great risk for COVID, so she's been very cautious. She's gotten pneumonia many times. She has to be on medication and regulate what she eats and such. It's not cystic fibrosis, but it is a error in that specific gene. She has that gene mutation, but it's not in the same spot. And so her impact on her cystic fibrosis gene, CFTR gene, is different. So I wanted to show you this picture because it relates to what I'm about to assign you for next week, okay? Next Tuesday, we will be talking about cystic fibrosis. and its various gene mutations. So, here you have nucleus, you have the DNA segment, you have the RNA that's properly cut, you have it goes through the endoplasmic reticulum and is now this is where it's built, okay? After it's built, it is edited in the Golgi apparatus and folded properly to become the mature protein it's supposed to be. So there are several different types of mutations. 1, 2, 3, 4, 5, 6. They all result in a specific kind of cystic fibrosis. So in one of them, the cystic fibrosis itself is not red. When it's red, it is a very short RNA, has one of those truncated nonsense mutations. When it goes to the endoplasmic reticulum, it is not red, and therefore there is no gene at all. Part 2 is the most common. What happens is that you get a full-length RNA, but the error of this phenylalanine means after it is red, that it's misfolded. And there's some protease destruction of the misfolded protein, and that either causes it not to be folded properly in the Golgi apparatus, or when it goes in, it's not properly it does not properly work. Okay, that's the most common. Okay, the phenylalanine 508 deletion mentioned before. That's the most common. 70% of all CF cases have this class of mutation. Any allele in that class that induces protease destruction is homozygous with any other allele of that class. But note that there are multiples even in that class. And note that any of these, so if one person has a allele that causes them to have absent functional CFTR because of this truncated RNA, and one person has this protease destruction, they still have cystic fibrosis. So having any one of these alleles can be, this is not the same as those completion concepts that we talked about. This is not a completion issue, because the completion issue is one of those pathways. The pathway for the blonde versus brown, that two people with blonde had different mutations along the pathway. This is not the same thing. Instead, any one of these alleles causes this impact, okay? You can have a defective channel regulation, defective channel itself, scarce functional, which is more similar to what my friend has, or decreased stability, making it so it's unable to, it has to be replaced constantly, okay? Again, my friend has more of the scarce functional, but she does have functional proteins. So this is an interesting piece. All right. So for the case study next week, I'm going to be posting it after class. You're going to be using this article. It walks through the different classes of mutations and how each one of them can cause cystic fibrosis. Because it's not as simple as we've made it out to be in terms of the very simple 1-to-1, 3-to-1 ratios that you expect. Gene mutations are myriad, because they can all have the same impact even if they're various mutations, which is intriguing. All right. So we're going to talk about single gene mutations and what's called beta thalassemia. So this is another kind of case study that's in your book. Sickle cell anemia is a single gene mutation, and it causes that single flip, causing one single missense. So a specific base pair change within the gene. B thalassemia is what's called a monogenic disease. It's an autosomal blood disorder. Now, the crazy thing is that there's many mutations. 400 different mutations in the HBB gene can cause the disorder. Two people who are carriers of two different mutations can still get HBB because they all affect the beta globin gene exactly the same way. Most cases actually have 20 separate mutations. So there's some crazy mutations going on, and all of them are affecting it the same way. And again, you can have one mutation, you can have 20 mutations, you can have 400 mutations, and they all end up with the same result. So again, there's this 5' upstream region, okay? So it has this transcription start site issue. Something to note is that even an error in the promoter can cause the lack of a specific protein, okay? So, for instance, there's 22 mutations known for this region, okay? 22. It's a lot of mutations. This is not just one allele. Every time if there's a new mutation, it's a new allele. But these single base mutations occur between this region, okay? And sometimes there's a T to A transition in the TATA sequence. Remember that TATA box regulates, is a core part of the promoter that indicates where the RNA polymerase is supposed to attach. If the TATA box with the promoter is damaged, this results in a greatly reduced transcription, and suddenly you have much less of that gene, even though the actual genes itself are present. And this is one of those things that we were talking about with the treatment for some of these. Something like this could be treated potentially with an activator, or with some sort of being able to put a segment of promoter back into where it belongs. This one, the cap site, the single base pair mutation, it leads to decreased levels of mRNA just because it makes the mRNA more unstable and it doesn't leave the nucleus as much. So again, this is outside of the translated region. This is the untranslated region that occurs on mRNA. When it is there, these single base pair mutations can cause, again, a decrease in transcription, decrease in translation. This is mild disease. This is the more severe disease here, okay, because the promoter's just gone. And then this one, the ATG translation initiating, the issue is that it alters the AUG site. Remember that AUG being the very important start codon. Translation doesn't happen because there's no AUG. It instead says ATG, and that's a severe disease result because you have no functioning protein, similar to the almost no gene transcription. So this is a fascinating piece, is that all of these mutations can cause thalassemia, and often people have multiples of these. So this is not a single issue. One single gene may be responsible for all of these, but the different segments cause expression differences when you have a mutation. Again, here's even more of them, okay? You have the exons. The disease severity varies depending on which exon is spliced out. You have the introns. It makes it so that the splicing doesn't happen, and that causes severe disease because you have a broken protein now, a very long and not folded properly protein. The polyadenylation site, that's the part, the 3N tail that protects it. It means that mRNA can't cleave and is unstable, but it can still often get out. And then again, there's more than 100. All of these other things. Insertion, deletions, duplications, etc. No one person is going to have the same combo of mutations, but everyone who has it on this gene. But it where it is and what kind of mutation is on the gene depends on the severity. So this is another part of why genetics is much more complex. We worked on so many simple problems. If your mother has it and your father has it, then you have a 50% chance of having it. But that's not true. You have a 75% chance of getting at least one of them and a 25% chance of getting two. But it's not fully true, right? This is the issue, is that if your mother has one type and your father has one type, then you could have it, even though maybe they don't even have any family history of it. Perhaps actually the mutation occurred occurred in the egg or sperm that made you, and you don't know it. So another interesting gene mutation I'd like to go through as well is this expanding nucleotide repeats. So what happens here is that instead of having just one CAG, which is this GIN amino acid, suddenly it has 3 copies of it all together. Can anyone tell me why might that happen? What's happening in the DNA itself that makes it skip like a record? Skip, 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 skip. What's happening? Anyone have any ideas about how this can happen? Again, it always happens with these trinucleotide repeats. Something to note is the trinucleotide repeats always include things like C and G that pair with each other. What could this cause? Any ideas for in terms of DNA processes? When the DNA is being replicated, what could be happening here? Ideas? Nobody? All right. We're a little lost, so I'll walk through it. But the concept here is that this is actually a DNA replication issue. This is not an issue of a random flip. These 3 repeats are for a reason. So, let's say this DNA molecule has 8 copies of a CAG repeat. Maybe that's part of the gene, okay? Q strands replicate and separate and replicate. With so many CAGs, you get this flip where the C bonds with the G bonds with the C bonds with the G bonds with the C, and then you have this hairpin for one of the CAGs. Now you have slippage here. It specifically happens with ones that have pairs on either side, okay? The CAG, when it flips down, the G and C bond and the C and G bond. And this one doesn't bond, but that's enough of it. So if it slips, then it continues there's no way for a DNA molecule to check. Wait, didn't I already read this? Because DNA molecules are not sentient here. The concept here is now you have more replications. When and then the 2 strands of a new DNA molecule separate. Now you have, and when they separate, so does this hairpin. Now it has 13. And if that slippage happens again, you can add more repeats. And if the slippage happens again, you can add more repeats. And this can be dangerous to stability of DNA replication, and any genes around that area with the repeats can cause unstable proteins. All right, so something to note is that slippage happens in the normal range. For spinal and bulbar muscular atrophy, there's a repeated CAG, like I mentioned before. In a normal person, you can have anywhere between 11 and 33 of these repeated repeats, these repeated triplets. In the disease, though, there's 40 to 60. So it's a little bit more than over the top. Again, there's a huge range. Fragile X can have a 6 to 54 of the CGGs. okay? But the people who are normal have this, the Fragile X has 50 to 1,500. That's a lot. So something I'm going to note here, we're going to talk about fragile, but you see that in a normal person, there's some slippage. In disease-bearing people, there's a lot of slippage, and it gets to these ridiculous numbers, up to 3,000 for the myotonic dystrophy, 3,000 repeats of the CTG, making nonsense and useless proteins that clog up the works, basically. You don't have functional proteins for important pieces. So this shows how this repeat expansion issue. Okay, the repeat expansion on these heavy repeat expansion on the X causes that fragile X issue. Okay, so normal control people have some variation in their trinucleotide repeats, but the three affected persons, have one X that is good, and ones that are really high. And this is the disease level. So this shows this person who's affected has a ton of repeats. This person who's affected has a ton of repeats. And the people who are related to him don't have that many repeats. Okay, so it's a complicated piece, right? Those repeats being caused. This is a trinucleotide repeat expansion pattern. Okay, that causes Fragile X and is partly due to these big segments of hairpinned DNA that keep over-duplicating. Diseases can result from mutations, including repeat expansions, even if they occur outside exons, because sometimes that means that the introns are not properly clipped because they are so long. And so all these occur in the introns. So while we might think of introns as junk DNA, even having errors there can cause the rest of the gene to be broken. So this is an interesting piece in the complexity of gene expression, is that depending on how many of these repeats you have, the repeats can impact the ability of the intensity of a specific disease. The different mutations can affect the intensity of the disease. So that's pretty interesting. All right. I'm going to see if anyone's actually in the chat right now because I haven't heard from you guys in a while. Which change is a transition-based substitution? You'll have to remember what the transition versus the transversion was. What's the transition? Guanine replaced by adenine? Yes, that's the concept. Is that it's a purine to a purine, okay? If it's a pyrimidine to a purine, then that is a transversion. Okay, good. All right. So we're going to talk a little bit more about the phenotypic. So this was the molecular impact. What does it do? But we're going to specifically talk about now how these impact the protein and impact phenotype. Okay, so we have a missense, nonsense, silent, and neutral. Okay, again, neutral, no change in function is not the same as silent. Silent is a codon to a synonymous codon. A neutral can literally just be no change at all. If a mutation that just changes one, like a single point mutation happens in a intron, that will be a neutral mutation. That is not just a silent mutation, okay? All right, so we're going to talk about the different pieces here. There's things called loss of function mutations, the reduction or elimination of a gene product, okay? As I mentioned, that's the elimination or the ones that affect the actual promoter period of the RNA. The null mutation results in complete loss of function. Basically, no if it's, let's say it is the promoter, the null mutation would be more, there is no mRNA produced. There is no protein produced. Maybe the AUG is missing, okay? So these are often loss of function or null mutations are recessive, okay? Because ones that provide function are often, the ones that provide function are often dominant. Ones that take away function are often recessive. So this is actually the difference between dominant and recessive. We've talked a lot about dominant and recessive and their impacts, but the key here is really a functional versus a non-functional protein. And is there enough being produced by one copy of a functional protein that it can override the impacts of a non-functional protein? If not, it's an incomplete dominance. So, for instance, my skin color is a conglomeration of multiple incompletely dominant genes that do not deposit melanin in my skin. I have melanin overall. I just don't produce it well in my skin, and that's several recessive mutations, loss of function mutations. A dominant negative mutation. So it's one allele may encode an inactive gene product that interferes with function. Who can tell me something we learned about already? A dominant negative mutation. Those are rare for sure. Something that's dominant and something that's also negative. Perhaps something that builds up over time and you don't see effects of it until you're older. You can think of a dominant one. There you go. Huntington's disease, yes. Huntington's disease is a dominant negative mutation. One of the keys here is that one allele, the dominant one, encodes an inactive gene product that clogs up the works. Even though there's one active gene product, the inactive gene product stops the working gene product from doing its job. That's how it becomes dominant, in the sense that it's fighting with the good gene. Instead of just losing a portion of the gene here, you're just not producing the protein, or the protein is completely non-functional. The key is that the non-functional protein that's being produced, if it interferes with the working of if it interferes with the job of a working protein, then you have that function interference and you have a dominant allele, even if you have one recessive that's working properly. And that's why Huntington's doesn't show up until later in life. You have one copy that's producing inactive gene products that get in the way of proper neuron functioning, and that slowly builds up over time. You can delay that, as we mentioned in the gene therapies part. You can delay that by reducing the amount of the bad Huntington's from being translated. So this can be haploinsufficiency, or it can be that buildup. Okay. Mutations can also be classified as gain of function or suppressors. If it results in a gene product with enhanced, negative, or new function, the key here now is that it's not a protein that is completely broken and non-functional. It's a new function. Then that often ends up being dominant, because instead of losing a gene product, you have created a new one that works better and potentially is more functional than the previous one. So this would be something, these are the kind of mutations that occur rarely, very rarely actually. Most mutations are detrimental. And I mostly talk about them as detrimental, but note that mutations are an important part of evolution. Sometimes you hit on a winner, that gene product that has enhanced activity. People who are able to process a specific toxin faster than others and survive better because of that. That is a gain of function mutation. and that can be dominant because of it, because their new gene product works better than the old gene product. So all they need is one copy of it. Suppressor mutation. This was an interesting one as well. Which one are you talking about? The Ebere. Which mutation are you asking about? Sorry, I looked away from the screen. The loss of function, the gain of function. So the gain of function one's actually interesting. It's not a new allele of the same gene. You actually just gave it a new gene altogether. Okay, it's not gain of function. The key here is that it is the UV bacteria. You actually just gave it a new plasmid. Bacteria are different than us, right? They can use those plasmids for non-functional pieces, and they can create the proteins from it. The plasmid itself isn't incorporated into the chromosome, but can be read by the bacteria. No, it's not like that one. If it was, that instead of giving them chromosomes in bacteria, if they are diploid, they do have two copies. Bacteria a little confusing, so bacteria probably a bad concept here. But if it was something where we took a jellyfish that didn't glow, and we took one of its chromosomes and replaced the old gene that doesn't glow with a new glowing gene, that would be a gain of function. That's a little bit different. The UV bacteria in the lab is more similar to gene insertion. We literally gave it a new gene that it never had before, not changing the allele of a gene it already has. Hopefully that explained it well enough. All right. A second mutation, this is an interesting one. This is a second mutation that reverts or relieves effects of a previous mutation. It can either occur within the same gene, perhaps a new allele can revert it back to wild type. We talked about those wild type versus mutants. Or it occurs in the genome and it produces a new product that changes the effect of the previous product. So let's talk about that a little bit, okay? So here's the concept of the loss of function mutation, okay? So loss of function mutation is when if you have two copies, you're producing a large amount of this product, one copy, a smaller amount of that product, and then if you have no copies, two mutants, there's just nothing. Or that loss of function where you have very few. Again, the intensity of cystic fibrosis is affected by whether it's a loss of function, this leaky loss of function, reduction in beneficial cystic fibrosis versus a lack of. Again, the gain of function is this. You have this mutant that's producing a new, better, or some sort of change in function, and that can override the previous function. All right. So these forward mutations versus reverse mutations versus suppressive mutations, okay? So you have a genotype wild type, red eyes. You have then you have a mutation that causes a dominant mutation that causes this this forward mutation changes the wild type into a mutant. Okay, so you have this dominant mutation that's added, white eyes suddenly. And then sometimes there can be a reverse mutation. If a mutation happens in the exact same spot, you turn A into a T, then you turn it back into an A. That's a type of intragenic suppressor mutation. Okay, suppressor mutation can happen again intra or intergenic. We'd call it a reverse mutation if it happened at the same site. Literally, it just changes back what was lost already, back to wild type. Suppressor mutation often refers to something that happens in another part of the genome. Basically, it's at a site different from the original mutation. It produces an individual that has both the original mutation and the suppressor mutation, but the suppressor mutation produces enough of the red eyes to act like wild type. So these two look the same, but they have a completely different genotype. again, this new mutation in the B causes the red eyes to come back, even though this mutant here has no red eyes. So let's look at the difference between the intergenic and intragenic. Again, both of these, so the reverse mutation and the suppressor mutation can both be referred to as suppressor, okay? Difference is that reverse mutation is the suppressor mutation that occurs in the same gene versus a different one, okay? All right. This is a mutation that hides or suppresses the effect of another mutation, hence the name suppressor, okay? All right, so you have the wild type. It has this active protein complex that has these two parts that click together, okay? The first mutation changes this gene, and you don't have an active protein complex, so now it doesn't fit. Second mutation in the second part makes this new part fit with the old part, and now it has the same function. If the suppressor mutation happens by itself, it will cause a different mutant phenotype. Possibly, well, this would be the same mutant phenotype because these still can't work together. They still can't join to each other. Again, the suppressor mutation only works if it's this suppressor is changing back to a wild type, even though technically the phenotype is different. The proteins are different. So intragenic, again, the second mutant site could be the same gene as the first mutation. You get this wrong protein now. It's a mutation, missense. Single codon turned into AAA, UUU. Now you have, instead of leucine, you have phenylalanine. The intragenic suppressor mutation, it can happen anywhere in the triplet. It doesn't have to be the same triplet being altered twice. Instead, the second mutation can occur at a different site in the same gene, but it has the ultimate same effect. UUA and CUU both code for leucine. And so even though those a different site, and this is a different codon you get the same result, and it reverts to wild type. We probably have tons of these in our own genetics that we don't know about. All of these changing and changing back errors, that's kind of why we have genetic variation. There's hundreds and hundreds of alleles that vary slightly that have turned on and off and on and off and on and off over generations. So we have complexities of codes inside of us. All right, so here's a little bit more of this intergenic suppression. You have the wild type sequence. It's translated, you have the leucine. If it makes this stop codon case, that's the nonsense. You have short non-functional. But site 2 encodes tyrosine tRNA. But what if it has an error that now makes that tRNA bring the wrong protein to it? It pairs with the stop codon instead when it shouldn't pair with the stop codon, but that means that translation it continues and you have a full-length protein. Again, it can happen even with the tRNA. So there's some intriguing pieces here that can work together in your body. All right. So one more check here, and then we'll wind down. Don't remember how much more of this I have, so I can't remember if we should stop it now or not. So let me look at it. So which type of gene mutation in protein coding gene has the least severe phenotype? Which one is the least severe in terms of functionality? Usually. Okay, we talked about some parts where these could be not as bad, but usually one of these is not as bad. Okay, Mavis thinks C. Anyone else? Ifania agrees. C. C. And this is true. Okay, good. So the base deletions, remember, can cause that extensive missense frame shift issues. Nonsense substitutions, that means you can have a shortened truncated protein. Expansion of a trinucleotide repeat, basically these useless proteins that are long and filled with a single amino acid. Okay, also unstable mRNA. All right, so let me see how much of this I have left. Probably get through just this piece, okay? I'll talk about spontaneous versus induced next week. Well, that's probably a good plan. Okay, so conditional mutations, right? These are expressed only under certain environmental conditions. That's what I have, right? These conditional mutations that are affected by environment, right? So this mouse carrying this gene controlled by tissue specific promoter, conditional genes, okay? And so this means that specific individuals can will have it or not have it, even if they have the gene. Then we have the lethal mutation that can result in the death of the cell. Okay, so these that was what we looked at in the mouse. That lethal mutation with two of these, gene A and gene B, together can cause a lethal. And those combos together meant that you had the loss of certain individuals if they had two copies. All right, so here's our final concept check, and then I'm going to show you a big table that will help you to keep track of all these, okay? Mutation in gene X overrides the effect of a previous mutation in gene Y. It restores wild type phenotype. So what is a mutation in gene X called? Everybody thinks it's A. Fania thinks it's A. Anyone else have an idea? You guys have a minute to decide. All right, well, I'm sorry, but it's not A. We have a C. Anyone else have more ideas? Kim thinks it's a C. Mavis thinks it could be a D. So Mavis, I'm only going to say the D indicates there's no effect for a neutral mutation. Since there is an effect, even though it's not detrimental, it is not a neutral mutation. Kim's got it. It is C. The key is that a reverse mutation usually happens. A reverse mutation specifically refers to the same site on a gene being altered to create the same, to just revert to wild type exactly. So they have the exact same DNA sequence as the wild type. Intragenic, that one changes in a single gene. In a single gene, it can change a different site to revert to wild type 2, switching a different nucleotide site to cause the previous wild type amino acid to come back. But this is an intragenic one because gene X overrides gene Y. So it would be C, intergenic suppressor, because one gene affects another gene. And it overrides it and returns it to wild type. That was a previous one where realized that the mutation in gene X by itself would not override. E itself is just a forward mutation that changes it from wild type to a mutant. Okay, so I'm trying to think of how I can type this out. Let me pull this up real quick. Pull up my notepad. All right, new note. No, I don't like this. It's too much personal stuff in my notepad. Don't look at that. Okay, so let's just pull up my Word documents here. Okay. I'm just going to type this out so you guys have an idea. Okay, so let's go up to 200. Okay, so forward mutation. GAG, GTG, mutant phenotype. This is the forward, okay? Reverse. Specifically, the reverse changes that. Is my screen blurry? Can you guys not see me? Let me look and make sure what's happening here. I can see it on Twitch, so hopefully you guys can too. This is blurry. You guys can't see it very well. Okay. Maybe it was just a glitch in the internet. All right. Again, this here. Go here. This creates a wild type. Okay. So forward is this, reverse is this. Reversing the same exact thing. Okay. All right. So forgive my, I would have to look at the amino acids chart, but let's say that a intragenic would make a GTG to, let's pretend that GTG and GTC, okay, see how it's a different site here that's altered? But that would revert it to wild type. Do you understand? Okay. Finally, we have the intergenic is the GTG, that mutant phenotype, impacted by tRNA mutant bringing original. So this is one way it can happen. This is a concept here, is that the tRNA mutant is a different gene. tRNA mutant bringing the original, what would be the CUC amino acid. Okay, does that explain what I was talking about? You guys get what I'm saying here? Intergenic, again, is two different genes working together to revert to wild type. It only works for the mutant. The reverse is literally the same location, switching back. Intragenic is a different site changing, but the result is the wild type. And the intergenic is where two different genes work together to revert to wild type. So in that sense, the intergenic would be the, let's say the brown hair would be wild type, blonde hair is a mutant, and the ones that we were talking about with those two people who had different alleles in different parts in the pathway to make brown hair, repairing each other and creating a wild type, brown hair wild type. Okay, so that's the concept. Okay, that's all I have. I will pull up this final giant piece here. Okay, so hopefully look at this table. This is from a different book, but it does outline all the pieces that we talked about, okay? 10 minutes, we're going to talk about we could talk about the exam if you'd like. Oh, it looks like I'm having some trouble with my framing here. Hold on. Ooh, yeah. Okay, I am pretty glitchy here. So I can see that, okay, looks like I'm back to normal, but my internet is having a little bit of issue. Now it says I'm super great with my kilobytes per second, but it says 0 for a second, so I apologize if I cut out. Good news is that I can watch that go up and down, so if I can see it happening. Okay. I apologize if I'm glitchy, but I hopefully this has passed. We just had a big wind go through, so maybe that impacted the connection I had. 10 minutes break. We'll come back and we can discuss both the test if you're still confused about pieces, and again, if you've already taken the test, do not say anything. And then we'll also discuss um, the homework. Okay. All right. I'll turn off the stream for now, and I will. I'll pull up the homework. Okay. And again, I do have the cystic fibrosis, the various gene mutations case study ready. I was finishing it up when it was time to start lecture. So I'll see you guys in a few minutes. Okay.